Hi everybody, today we're going to talk about DNA structure and function. This is our first guiding question, first lesson. I want you to take a good look at this picture here. This is DNA, it's a double helix, and it looks like a ladder, but it's a twisted ladder, and that's going to be important as we go through here because we're talking about structure and function. What is DNA? DNA is the genetic material in cells, and it is contained in a molecule. All the genetic material in cells is contained in a molecule called deoxyribonucleic acid, or DNA. It determines the body's structure and function. So everything about how your body works, what you look like, some of the illnesses, or maybe if you have asthma, or if you're predisposed to diabetes, all that would be in your DNA. We were talking yesterday about those who had receptors that were closed, so they were not susceptible to the Black Plague. That's contained on your DNA as well. It's found in the nucleus of the cell, so those of you who are 8th years, remember last year's we talked about cell function and structure, that the center of the cell is the nucleus. 7th years, you've probably learned this too. And inside that cell, in the nucleus, the DNA is protected. So it's kind of in a double lockdown space where it's protected. DNA is very individualized too. So all of us sitting here listening to this right now have an individual specific DNA. That's how science is amazing. Our crime scene investigators have figured out how to use DNA to help solve mysteries and crimes. So because it's individual, like a fingerprint, that can help us in situations such as a crime scene. It can also help us to show people are innocent too. It's shaped like a ladder. It's a double helix. So we've got this ladder, but not only is it a ladder, but it's a double helix. So you've got these bases that run down each side and it's twisted. Think about like something that compacts, like a spring or a slinky, how it compacts. It's really, you could stretch it out to be quite long, but it also comes together really small. So that would be your DNA. In fact, you have so much DNA that all the DNA in your body could reach from the sun to Jupiter and back again more than 8 times. So your DNA is compacted quite a lot in your cells. And it's very small. How was DNA discovered? Well, we've learned a lot even in my lifetime about DNA, and certainly you'll be able to say that as well. In your lifetime, we've discovered so much about DNA. You've watched the Bill Nye video where he goes through a lot of the people who contributed to our understanding of DNA. And that's because today, and that's still growing, today lots of scientists from all over the world are still contributing to our understanding, and lots before us have contributed to our understanding. Some of those scientists discovered its chemical makeup, others learned about how the chemicals fit together, still others learned about its shape. That was a key. So these are a few of our players, important pieces in the DNA timeline. In 1857, we have Gregor Mendel, who did the pea experiments. So he was a monk in a monastery and had a beautiful garden. And in that garden, he just had some common pea plants and started doing some work on genetics. And he made a lot of discoveries in his garden. And then in 1869, Johann Friedrich Michener, he isolated DNA. So this is something that we're going to do with simple things that we have just in our homes. We can isolate our DNA. We can extract our DNA. But that's because we stand on the back of giants like Friedrich Michener, who figured it out before us. In 1919, we have Phoebus Levene, who has figured out the bases, an A base with a T base go together, and a G base and a C base go together. So we're showing here how these sort of fit together in one another. They have sort of the same puzzle piece. In 1928 was Frederick Griffith, and he figured out that DNA can be transferred. So if you cross 2 pea plants together, you've transferred some of that DNA. It can be transferred from a parent to an offspring as well. In 1948, we figured out, 1948, guys, that is not that long ago, that DNA controls heredity, meaning it can be inherited. So from 28 to 40, that's 20 years. We figured out it can be transferred, but didn't figure out it could be inherited until 20 years later. And then we have Franklin Wilkins, who took an X-ray of DNA in 1951, and we have a visual now. And then 1952, Hershey and Chase worked with viruses in DNA. So this is what a virus looks like. It has like this little injector that comes down and injects more copies of itself into your cells. This is something that if you're interested in viruses and how viruses work, especially in the day and time we live right now, this is one of the choices, and you can look into how viruses work. There is great debate still in the scientific world of whether or not viruses are living things, because they can only live when they have a host. So is that a living thing? No, you can look into that. In 1953, we have Watson and Crick, who came along and figured out that it was a double helix shape. So let's look at that one. What does DNA look like? The two sides of the ladder shape are made of sugars and phosphate groups. So here you can see there's a sugar group and a phosphate group, alternating patterns, right? Those are the sides of the ladder. The rungs, so the middle pieces, are the DNA base pairs. So a C matches with a G and a T matches with an A. A C and a T don't go together, and an A and a G don't go together, just like an A and a C or a G and a T would not pair together. It's like pieces of a puzzle. So here are our base pairs. Okay, this is called a nucleotide when you have sugar phosphate base there. And then this is the double helix, so this is what it looks like, kind of uncoiled. And then this is what it looks like, the DNA coiled around a protein. So here's our DNA, this purple is coiled around this protein. Look how tightly coiled it is. You can't even recognize that it's the shape, and then it comes down to this. It reminds me of when we discovered molecules, right? Or elements. We just thought that the wind and fire and air and water were elements. Then we've discovered this entire periodic table of elements. So at first, this doesn't even allude to the amount of detail that all of this shows us. So we've come a long way in our understanding. The building block of DNA is called a nucleotide, which is a base and a sugar and a phosphate group. This is a nucleotide, a base, a sugar, and a phosphate group. And these are, sugar is this shape because of its molecular structure, sort of builds this shape. So a base, a sugar, a phosphate group, a base, a sugar, a phosphate group, and so on. Okay, that is a nucleotide. There are 4 different nucleotides in DNA. We have an A base nucleotide, a T base nucleotide, and these two pair together. We have a G base nucleotide and a C base nucleotide, and G and C pair together. So they are paired. Adenine always pairs with thymine. These are amino acids. So adenine always pairs with thymine. So that's the AT group. Cytosine always pairs with guanine. I'm sorry, it's cytosine. Cytosine always pairs with guanine. So there's the G and the C. The order of the nucleotides in DNA is a code that carries information. It determines which proteins should be built. So let's say you have a string of 4 CG, 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 CG. That tells our cells to make a certain protein, and that certain protein then is shipped out to our body to do a specific job. A scientist determines that a molecule of DNA is 27% cytosine. What else does the scientist know about the DNA? I want you to pause this video, and I want you to try to figure out what else. Think about the base pairs and what else you could know if you know that a molecule of DNA is 27% cytosine. Pause now. So the scientists would also know that guanine is 27% because C and G always pair together. So if there's 27% cytosine, there's 27% guanine because they're always paired together. So how are copies of DNA made? This is where we're going to start to get into some technical information. So if you need to stop, pause the video and do a Google search on your own to look up more information, do that. If you need to start an email and writing me questions that you have, do that. In replication, so the cell makes copies of the DNA molecules through the process known as replication. And during replication, the DNA begins to separate. So what happens is the base on each side are used as a pattern for the new strand. So this begins to separate right down the middle. All of it opens up. It unzips, we call it. Replication proceeds and complementary nucleotides are added. So if this is an A and a T and it opens up, this is an A T will be added. This is a T, an A will be added. So this is unzipped. Think of it unzipping just like a zipper. Now, instead of the zipper coming back together, we're going to add a new side of the zipper to every one of these. And it's a complementary one. A G is not going to go with an A and a T will always pair together. When replication is complete, now we have two identical DNA molecules. So how is DNA copied? First of all, you have DNA becoming unzipped. All right, think of this like a zipper. There is an enzyme that attaches that does the unzipping. And then DNA polymerase attaches. So this is another enzyme that attaches to each side of the unzipped molecule. And then it starts adding down the complementary nucleotides. So if purple is an A and yellow is a T, you're going to get a yellow on every purple, okay, and a purple on every yellow here. So the nucleotides are added. You're getting your second identical strand. And then DNA polymerase detaches. So that molecule comes off. It's done its job. It has replicated all of the DNA, all of the bases, and paired with a complementary new one. And now you get two identical molecules out of this. So one becomes two. So this is a more in-depth picture. This molecule is unzipping, and this one is attaching, and you're getting complementary base pairs attached. Got my complement, and then the polymerase comes off, and you've got two new DNA identical strands. When are copies of DNA made? The 4-cell divides, it copies its DNA. So our cells can replicate DNA in just a few hours because replication begins in many places along the DNA strand. It doesn't have to start at the beginning and go to the end. It can start along that very long strand of DNA anywhere. And many groups of protein are working to replicate your DNA simultaneously. It's all happening together. So if you could look at this video, my shockwave isn't working, but yours may. And I will post this on Google Classroom today too. This is an opportunity for you to go and unzip some DNA. What is the role of DNA and RNA in making proteins? Well, I haven't even told you what RNA is. RNA is ribonucleic acid. So DNA is deoxyribonucleic acid. That means 2 oxygens. RNA is ribonucleic acid. So protein factory. To build proteins, some of the information in the DNA is copied to a separate molecule called RNA, or ribonucleic acid, and it is used to build proteins. I'll go to that so you can see that. RNA is used to build proteins. So mRNA is a messenger type of RNA, and it copies instructions from the DNA during transcription. So transcription, like a transcriptionist is a person who, like in a courtroom, just records everything, types up all the notes that's being said from each person. So think of that as you think about DNA transcription. So like DNA, RNA has a sugar phosphate backbone, and it has the bases adenine, guanine, cytosine. But instead of thymine, it has the amino acid uracil. And there are 3 types of RNA. Each type has a special role in making those proteins. So, when a cell needs to make a protein, it makes an RNA copy of a section of the DNA